Welcome everybody. This is our chapter 14 notes. This is gene to protein. So what we're going to talk about today is the idea of using our genes, our genetic information, to actually do something. So what is really going on is that these genes are coding for how to build your proteins. And you have tens of thousands of different varieties of proteins, and honestly what makes you unique is that all your proteins are slightly different from everybody else's. Nice picture here of essentially an albino deer. So what is happening there is that there is a mutation in one gene, and that gene is going to basically cause that deer to not have the ability to produce pigment. And so what ends up happening is the proteins that are necessarily make the color in these deer cannot be produced in this deer, and so you end up with an albino deer. So similar mutations exist in many different animals, including sperm whale. That whole idea of Moby Dick being the white sperm whale, it's a real thing. Not necessarily Moby Dick, but the idea that there were albino white whales. All right, so I'm going to really quickly zip through this idea of an experiment that was done. They found all these mutations in this one group of yeast, and what was happening is they were growing them without a certain amino acid. In this case, arginine. And you need arginine to survive. Every organism would. And what they were able to do was create a whole series of mutations, different organisms, these three classes of mutants, and then this is the normal one. So the normal yeast was able to grow even without any of the raw ingredients to make arginine. The class 1, as long as you gave it ornithine, it could actually produce these. And then class 2, you had to give it citrulline, and class 3, can't really see that, had to give it arginine. So what was happening here is essentially there was a metabolic pathway, and you can see it here, enzyme A, enzyme B, enzyme C, and the mutants were deficient in one of those enzymes. So you can see that this last mutant over here, the class 3, the only way it could actually survive is if it actually had arginine. So what this meant is this idea of, okay, genes are coding for proteins, which is true. And they came up, this experiment in 1941 came up with the theory of the one gene, one protein hypothesis. And unfortunately, although that sort of stuck around and people still use it as an example, it's really not accurate. Because the truth of the matter is that many proteins are made of multiple polypeptides, which means that there's multiple genes that you need for one protein. So now they sort of updated it, and they now call that theory the one gene, one polypeptide theory. Again, it's not completely accurate because you can actually take a protein and edit it slightly different, and you can produce different proteins. So the modern theory is really kind of a hybrid of all of that. I just made that up, the mostly one gene, one polypeptide. So, but essentially what we're talking about here is this gene, this functional unit of DNA, codes for some part of a protein. And in some cases, one gene, depending on how you put it together, could actually produce a couple different varieties of protein. I'll give some examples later on how this works. So here's the basics. And some of this you should remember from 9th grade biology, but what we have here is the idea that we start out with DNA. DNA codes for everything. Every piece of information you have is in your DNA and your nucleus. You don't need it all the time. So, for example, you produce a protein in your eye that is able to sense light. It's the key to your ability to sense light. That protein is only produced in your retina. It's called retinol. And although every cell in your body, your liver cells, have that gene inside the cells, they obviously don't make the protein, though. You don't need it. So the DNA is sort of the master plan. It has all the information, but you're only going to pull the genes that you actually need at that time to start producing them. In order to do that, we need to take that DNA and make a copy of it, a working copy of it, and that's where we make the RNA, the messenger RNA. And we then use that messenger RNA to actually go and code to make the protein. So we'll go through that process. So basically, the process of going from DNA to messenger RNA is called transcription. You probably heard that term before. Transcription is, transcribing something is to rewrite it. Or if you're taking, if you have a transcriber in court, they're taking what is said and they're actually going to write it down. So what we're doing is we're taking the DNA piece and we're making a messenger RNA piece out of that. There's this intermediate step called RNA processing where we actually take that initial transcript and we actually edit a little bit. We cut it down, we add some things, and there's some tricky parts about that. And we're still not 100% sure about everything with that. That's kind of a newer thing that we've discovered. And then the last part is translation. That is where we take the RNA and we turn it into an amino acid sequence, and we use ribosomes to do that because ribosomes make proteins, and you know that already. And then we have the last step is to fold up that protein, because remember, just having the series of amino acids is not the final protein. It's got to be all folded up, all those different levels, those primary, secondary, tertiary, quaternary structures of proteins all have to come into play. Making that first translation, we're only going to do primary structure of a protein. So, and last here, the RNA processing step, this step in the middle here, is only happening in eukaryotic organisms. It does not happen in bacteria. So yeah, just a quick idea here. This is bacteria. We skip that RNA processing step. And then this is eukaryotic organisms. So just subtle difference there. All right, genetic code. So as you, I'm sure, probably are already aware, we actually read DNA in groups of 3 letters. That nucleotide sequence has to be read in 3s, and there's a clear and obvious reason there. We only have 4 letters, A, T, C, and G, or A, U, C, and G if we're talking about RNA. If we only read 1 letter at a time, there's only 4 possibilities, and that's not enough to code for the 20 amino acids. Even if you put 2 together, AC, AU, all those, there are only 16 possible combinations. That's still not enough to get all 20 amino acids. So we have to do it in groups of 3. And that gives us way more than we need. We actually end up with 64 possible codons there. And the next slide actually has all of them listed. We have 1 start, we have 3 stop codons, and then anywhere from 2 to 4 different sequences that code for each amino acid. We need the starts and stop. Very much like we read sentences, we know that we put a capital letter at the beginning of a sentence and we put punctuation at the end. That is why we have that start and stop. It provides what's called a reading frame. So you can actually know, okay, we start here, read 3 letters at a time, 3, 3, 3, 3, 3, and then, okay, stop here. That's the end of the gene. And many of the codons actually only vary on their last base. So what that means is that there are some codons that the first 2 letters are all you need, because it doesn't matter. The 4th letter is always, it's always going to end up coding for the same thing. And perhaps one of the most remarkable things in biology, I think so anyway, is that genetic code is universal. You basically can take the gene from any organism on the planet and put it in any other, and they will make the protein. It's really amazing. is a clear indication that all organisms evolved from a common ancestor. And it's also made for some pretty crazy things that I'll show you on the next slide, that we now have this ability to take genes from one organism and put them in another. And one other little tidbit here, when you're actually looking at the DNA, even though there's a double strand, there's two strands to that, only one side of it is actually the coding strand. The other side is often referred to as the non-coding or nonsense strand. And then you have the template strand, or the coding strand, is the one that actually provides that. Now, for different genes, that coding strand can be on one side or the other, but within a gene, one gene, there's only one side of the DNA that actually codes for something. There's the code. You've probably seen this before in 9th grade bio, and you can see, for example, proline here. Anything that starts with CC, it doesn't matter what the third letter is, it's going to still code for proline. And those are all the 20 amino acids. This is our start codon here, and then our 3 stops there. Start codon does code for an amino acid as well, and the stop codons do not. This is what we can do. This is a tobacco plant with a firefly gene in it. This is a pig with a jellyfish gene known as the green fluorescent protein. There are so many organisms out there with what's known as a GFP. You can go to Q-Mart, not right now, but you can go to Q-Mart and you can get a goldfish with the GFP gene in it. They pretty much put that gene in everything. It's a really common one to use. All right, so how does this happen? So transcription, it takes place in the nucleus because that's where the DNA is, and we need the DNA to transcribe. It starts out with these things called transcription factors. It's complicated, but you basically bind these transcription factors to an area called the promoter region, which is right in front of the gene, right at the beginning of the gene. The promoter usually contains these things called a TATA box. That sounds really weird and silly, and it is, but the reason they call it that is just it's a whole series of T's and A's, and the reason that is significant is when you have a series of the DNA that has T's and A's, it's a little weaker and a little easier to unzip. So that TATA box is, you know that when you have the bag of chips, there's that perfect spot you can pull where it's really easy to open it. That's the TATA box. It's a really easy part of the DNA to unzip. RNA polymerase II, you've heard the polymerase before, very similar molecule, but in this case it's obviously going to be reading the DNA and writing RNA. And then you have this, what's called a transcription initiating complex. It's a very large structure, complex, proteins, and a bunch of other things. And prokaryotes don't actually even have those transcription. But once you actually start the RNA polymerase, it starts transcribing, moves 3 to 5 down the template strand, meaning the RNA strand is built 5 to 3, the opposite because of the anti-parallel nature of DNA and RNA. And then eventually you get to a termination sequence, and at that point the RNA breaks off, and you now have a pre-messenger RNA. That's what this means right here. It's not quite messenger RNA because there's a couple other steps there. And then we don't really understand exactly how that RNA polymerase falls off, but it does, and we're ready to head to the next step. This is showing you that TATA box. This is those transcription factors. And once you actually write it, start transcribing the DNA. It's pretty straightforward because you just write the opposite. Notice in this top strand here is the non-coding strand, and the bottom strand here is the coding strand. Bottom strand is being read 3 to 5 because it's moving in this direction, and the RNA is being written in 5 to 3. Okay, and then we eventually get to the end and we fall off. I'm going to skip to the next video to start the RNA processing, and we'll start there.